Hello, my name is Martin Reyes, and today I'm going to be talking to you about the ultrastructural causes of retinitis pigmentosa and the variations of gene and stem cell therapies used alongside with organoids. So I'm going to go ahead and split up my presentation into 6 different sections here. And we're going to go ahead and start off with talking about the background here, the retina, the anatomy of the retina. So the retina is a very complex tissue residing at the back of the eye, right? And it is encompassed by the choroid, which is a highly vascularized membrane layer responsible for the transmission of blood throughout the eye. The major role of the retina is actually going to be in the phototransduction, right? So that's going to be photons entering your eye and interacting with these photoreceptors and getting converted into a electric signal that will be sent to your central nervous system and interpreted as visual data. So several different layers here. We're going to talk about 3 of them of the retina. And first you're going to have your outermost, the retinal pigment epithelium layer, and this is going to provide a foundation for photoreceptors to reside on, and will actually also provide nutrients and promote the overall health of photoreceptors, including through methods like phagocytosis to relieve oxidative stress. So the next layer that you have are the photoreceptors themselves, and they're going to be in the outer retina, right? And then lastly, you're going to have the inner retina, the outermost part of the retina. And these are going to host the secondary neurons, like your bipolar cells, horizontal cells, amacrine cells. And this is where you're going to have your signals being sent to the optic nerve and out into the central nervous system. So here you have the vitreous, which is the empty area where the light's going to be kind of coming in. So retinal degeneration is going to be the degradation of the structure, and this is usually caused by genetic or environmental factors. So some examples are age-related macular degeneration, right, which is actually the leading cause of blindness in the Western Hemisphere. And you're also going to have diabetic retinopathy, which is going to be this abnormal growth of blood vessels that damage the retina. And also, we're going to have inherited retinal diseases, which we're going to touch on in just a second. I want to include this diagram because I really like it. It's very nice to look at and shows this cross-section of the retina. You can see what a healthy retina looks like, all your photoreceptors, all your neurons, and then you can see in different phases, and eventually it completely decays into this grotesque deformity of what its former self used to be. So I like this one. I wish I had included it into my report. So going back to inherited retinal diseases, right, IRDs, so they encompass a variety of retinopathies, and there's actually about 270, more than 270, right, 270 plus genes known to cause inherited retinal diseases, and we can see a few of them here and their respective genes. And the biggest one here, the biggest one of all, and the most common and well-known one, is retinitis pigmentosa. So that's going to be the focus of our report here. And retinitis pigmentosa is predominantly identified by the photoreceptor death, specifically in the rods, right? So that's what's going to be the onset of gradual retinal degeneration. And it's usually identified first in adolescent or teenage years. Now, usually at the time that at this time, it's asymptomatic. So oftentimes patients don't even know that they have it yet. And this is in spite of large amount of photoreceptor death. Even then, they still won't be able to notice that they have anything wrong with them, which eventually will lead to the first symptom of retinitis pigmentosa, and this is going to be night blindness. And this is identified in adolescence to young adults, maybe or teenage years, 20s. And the last 2 symptoms are legal blindness and complete loss of vision. And reports of this are usually in the 40s and 50s years old and can go on to the elderly age. So we can actually see here tunnel vision, which we're going to talk about in a later slide. So hereditary and variations of retinitis pigmentosa. So there's going to be 2 main categories that I found that distinguish retinitis pigmentosa, and you're going to have your non-syndromic variants, which are usually triggered by a single gene, and these are considered to be easier to identify because it's not from a whole source of problems. It's usually something malfunctioning, and it can be traced, versus syndromic variations, which arise from a larger host of problems, and this form of retinitis pigmentosa usually comes from a bigger problem. So an example of this, right? So here are your non-syndromic ones that we just talked about. And your syndromic ones, a good example is going to be Usher syndrome, which is the loss of hearing and vision in a patient. And this is because a protein, the USH2A, the usher protein and the USH2A gene are malformed, and this is what causes a whole host of problems. So first thing we want to touch on here is the actual cause of retinitis pigmentosa, right? We talked about that it's particularly identified by the death of photoreceptor rods. So we see here a photoreceptor rod, and we're going to be discussing about most classical forms, so like your autosomal dominant iterations of retinitis pigmentosa that are caused by this deformity in rhodopsin. And this particular protein is not only important in the initial stages of phototransduction, but it is also important in the development of the outer segment of these photoreceptors, like the most outermost part, the part that's hugging the retinal pigment epithelium. And so what happens is this loss of function with rhodopsin, and the outer structure, outer segment, is not particularly made well. This is what we'd consider a ultrastructural defect because it affects the fine structure of the photoreceptor rods. So this will lead to death, right? And the first symptom that we had talked about, night blindness, is actually a direct cause of this because rod photoreceptor cells are what help you see at night, right? They're usually they're adapted to help you see in low light settings. And so this is why eventually when you have so many of them die off, your visual acuity in these specific environments is significantly decreased. And then now we're going to talk about tunnel vision, which is the next symptom, and the one that defines retinitis pigmentosa the most, and the one you'll hear about. So rods in the retina significantly outweigh cones, right? Cones are usually condensed to just this fovea area. Rods are distributed all over. And what's going to end up happening is that some of your more outer rods, when they die off in significant quantities, the parts that are less rod rich are going to suffer significant damage, and that is what's going to cause your loss of peripheral vision. Now, next up, your complete loss of vision is going to be because now not only have you been losing your rods, but you're also starting to lose your cones. So at the point that retinitis pigmentosa spreads to the cones, you're in the end game of retinitis pigmentosa, and that's when people completely lose their vision. So retinopathies and IRDs like retinitis pigmentosa affect a large portion of the population of the world. And so there's active research going into different therapies and methods that we could do to treat these terrible diseases. And one of the first ones we're going to be talking about here is viral gene therapy. Now, viral gene therapy is going to actually utilize viruses to deliver drugs and treatments into the nuclei of affected cells. So it's kind of using the biological advantages that viruses had over millions of years of evolution. So the pros are going to be high transfection rate and high rate of host genome integration. And again, this is because viruses have evolved over long periods of time to be good at this. And now we're going to try to engineer them for the good of our cells. Now, there are some cons in that safety concerns. These are still viruses, and our bodies have immune responses to virus expression. And so also there's high production costs, low carrying capacities, which we're going to talk about in specific in just a second here. The main viral gene therapy we're going to hone in on are adeno-associated viruses, or AAV vectors, right? So here's a nice little picture of one here. This is going to be a protein shell with a small single-stranded DNA, usually about 4.8 kilobases, and it's a heavily looked at variant of gene therapy because it is known to be the safest version of gene therapy, and it's actually a very versatile method. And you'll see different derivations of the AAV vectors used for targeting a multitude of different cells. So that's the versatility, is that you can reprogram these things and structurally define them to latch onto cells that you would like to target. Now, the biggest drawback of AAV vectors are their small carrying capacity. So it's about only 5.2 kilobases, and we're going to see when we compare them to non-viral genes, that is a blow. to their applications. So pure non-viral gene therapies, right? This is the next one we're going to cover. So as the name entails, you're going to be using other molecules that aren't viruses as these drug vectors. So the pros are that they are very safe because they don't contain that genetic material we were worried about with the viruses that might trigger an immune response. They have a high carrying capacity and low production costs. Now, the cons are actually going to be what made the viral vectors advantageous, and that they induce transfection a lot better, and the target delivery of them was more accurate, as opposed to here, we may not get them into the right cells that we need them to get into to deliver their payload, right? So specifically, polypeptide-based nanoparticles have been gaining some favoritism in research, and this one in particular, the CK30PEG, theoretically has no carrying capacity limitations. There are several reports that have tested it at about 20 kilobases worth of genetic material, which is sufficiently better than the 5.2 kilobases that we saw with the AAV vectors. Again, high safety because they don't contain that viral DNA. And they were able actually to manage to get high transfection rates, even in systemic organs such as livers, such as lungs. They seem to have done pretty well in many different trials. So they're versatile and customizable. So they just have many benefits that the AAV vectors offered. And we can actually see here a table that shows all these different vectors, right, derivatives of the AAV, and then CK30PEG. They were able to target a good amount of retina cells, and their carrying capacity, of course, was significantly larger. And what this paper concluded was that their onset of expression was actually a lot sooner than most of the other methods, and this made CK30PEG a preferable therapy to look into. So next up, we're going to talk about induced pluripotent stem cells. Now, pluripotency is the ability for a cell to differentiate into other cells. This is kind of the hallmark of stem cell therapy, is the ability to have a kind of a blank canvas and force it to become whatever kind of cell you want it to be. Now, there are several approaches to this particular process, including embryonic, induced, reprogrammed, but embryonic stem therapy has been kind of shunned and looked at as from an immoral angle, typically because people are uncomfortable with the idea of using embryonic human cells to be experimented with. And so what ends up happening is that induced is a preferable method. So with induced pluripotent stem cells, what we have are adult somatic cells, so right, just your typical body cells. And with the introduction of these transcription factors, they revert into this pluripotent stem cell state, into which then we can introduce differentiation factors and make, like a erythrocyte or a neuron that we would need to make. Now, specifically relating this back to retina cells, right, we run into some problems in that trying to create the environment of the eye, that kind of three-dimensional structure, is nearly impossible on, say, a petri dish to get accurate kind of a retinogenesis of these cells in their normal environments where they would be surrounded by other cells with complex cell-to-cell interactions. And so researchers solved this with the development of organoids. And organoids, you can kind of think of them as simulations of these organs in that facilitate the multidimensional growth, the proper development of cell-to-cell interactions that you would probably see in a real human organ. Another benefit of these is that since we're growing them, we can actually grow them with patients' genetic material. And something that you'll see as problematic in, like, for example, blood transfers or organ donations is that the recipient will often experience immune responses to these and often rejection to some of these organs. And so ideally, if we're using the patient's genetic material, then this should mitigate a lot of those problems that we would have if we were to try to transplant cells. And so this is kind of deriving on that individualized medicine, which we'll probably see a large increase in interest in over the many decades. So the many applications of organoids, right? We talked about cell therapy with pluripotent stem cells. Pathogenesis is actually going to use these organoids to kind of simulate what a disease and how it acts in these human organ environments. So, for example, there was a successful implementation that I read about a RPGR mutation that was actually used to model the pathogenesis of that mutated gene, because up to that point, animal models had not been sufficient to replicate the same kind of phenotypes that you would see in humans. So organoids are pretty valuable in not actually testing and messing around with human organs, but those kind of simulated environments. And that actually relates to gene therapy, which we were just talking about, right? So this is going to be testing out different drug delivery methods, kind of like AAV, which I have a diagram here, and testing them out in these simulated human organoids. Now, there are some limitations to talk about as we go ahead and wrap up our talk here, and it's that they are expensive to create and maintain. As if that's not already enough of a burden and challenge, we also get to more complex limitations. So, for example, physiological and structural. So, one instance is the lens. You know, the lens is going to affect cells if we were going to try to grow different parts of the eye. The environment that the lens grows in is highly specific to the actual overall structure of the eye, right? So trying to replicate this in a simulated environment is pretty difficult. An even better example will actually be these blood vessels, right? Because in a regular human body, we have a circulatory system that's pumping out all this blood, sending it throughout the body. But in an organoid, we don't have that circulatory system. We don't have a pumping heart that sends blood all throughout the body. So simulating these blood vessels is also another challenge in trying to properly replicate what an organ would do. So that's going to go ahead and wrap up my talk here, and I'd like to say thank you to, of course, the viewer, you, for getting through the entirety of this video. I'd like to thank my professor, Dr. Rowe, from the University of Houston, the Department of Biomedical Engineering, and I'd like to thank you. Hopefully you have a great day.